What if the very story of your aging wasn't solely written in your genes? You know, like a fixed, unchangeable destiny. But what if it was instead a narrative that's constantly being edited, rewritten, profoundly influenced by the choices you make every single day?
It's a compelling thought.
Right. Imagine having two people born with virtually identical blueprints for life, identical twins. Yet over time, their paths diverge dramatically in health, vitality, longevity.
It does sound almost like a challenge to strict genetic determinism, doesn't it?
Exactly. Welcome to a deep dive into precisely that fascinating realm where our seemingly immutable genetics meet our incredibly dynamic environment.
It's the field of epigenetics.
That's the one. Today, we're going to unpack how our lifestyle, our diet, our stress, our surroundings don't actually alter our fundamental DNA sequence.
Crucially, yes. The sequence stays the same.
But rather leave these subtle yet really powerful marks on our DNA and the proteins wrapped around it.
Like little post-it notes or annotations.
Yeah, exactly. Like switches or dimmers influencing how our genes are expressed. They effectively change the performance of our genetic symphony and, crucially, determine how we age.
And for you, our listener, who seeks not just, surface-level information, but a profound understanding, actionable insights into this complex machinery of life. Well, our mission today is to thoroughly dissect this intricate dance.
The dance between epigenetic alterations, the cell's internal metabolism, and the overarching process of aging itself.
Precisely. We'll navigate the cutting-edge science, explore the most promising interventions, and also confront the critical questions that still remain.
Giving you a robust, sort of logically structured framework.
Right. To comprehend this complex field without feeling, overwhelmed by all the details. Our ultimate goal is to equip you with the knowledge to truly appreciate the subtle yet immensely powerful mechanisms at play in shaping your health span.
Health span, that period of life spent in good health, free from disease. Love that distinction. It's key. Think of it as peeling back the layers of a really sophisticated system. Understanding not just the individual components, but their dynamic, interconnected interactions that define our biological reality.
Okay, let's embark on this journey then. We'll begin by examining some of the most compelling, almost elegant evidence that really underscores the powerful role of environment in shaping our epigenomes.
The twin studies you mentioned.
Exactly, those fascinating studies involving identical twins. From there, we'll move into the revolutionary concept of epigenetic clocks.
Yes, measuring biological age.
Right, exploring how scientists are attempting to precisely measure our biological age, and maybe more importantly, delving into that critical debate. Are these clocks just passive markers of aging, or are they active causal drivers of the process itself?
A fundamental question.
Then we'll dive deep into the crucial crosstalk between epigenetics and cellular metabolism. We'll focus particularly on that vital NAD plus sirtuin axis.
And why its age-related decline, especially due to the enzyme CD38, is so significant. Yes. And finally, we'll broaden our scope a bit to discuss how other master nutrient-sensing pathways like insulin, IGF-1 signaling, orchestrate these widespread epigenetic changes.
And touch upon interventions.
And briefly consider promising interventional approaches, From the age-old principle of caloric restriction to the exciting new frontier of potential CR mimetics. Sound good?
Sounds like a comprehensive deep dive. Let's get started. # core content. # I. Epigenetic alterations and nutrient sensing crosstalk in aging. Twin lifestyles, epigenetic drift, and aging.
So we begin our exploration by examining one of the most compelling lines of evidence for this profound influence of epigenetics. Studies on monozygotic or identical twins.
Right. Individuals born sharing virtually identical genetic codes.
This biological congruence offers a unique, really invaluable natural experiment, almost like a controlled lab setting provided by nature itself.
It allows scientists to isolate and clearly observe the differential impact of environment and lifestyle on two genetically predisposed individuals. It's quite elegant, actually.
It's like having two identical supercomputers, fresh off the assembly line. And then watching how different software updates, usage patterns, maintenance protocols lead to vastly different performance, longevity, even susceptibility to glitches over time.
That's a good analogy. And what's truly fascinating and quite revealing here is that while young identical twins do indeed exhibit nearly indistinguishable DNA methylation and histone acetylation patterns at birth.
So they start out almost identical epigenetically, too.
Very nearly, yes. But a remarkable divergence, which scientists term epigenetic drift, invariably occurs as they progress through middle and old age.
Drift, so that's not random.
Well, it's the measurable, systematic accumulation of distinct epigenetic marks. It's not just noise. When we talk about, say, 5-methylcytosine DNA content, that's the most common type of DNA methylation.
Okay.
It involves adding a methyl group to a cytosine base, usually within specific sequences called CpG dinucleotides.
And this acts like a dimmer switch, usually silencing genes.
Generally, yes. It often turns gene expression down or off. Then you have histone acetylation levels.
Right, adding acetyl groups to the histone proteins DNA wraps around.
Exactly. Histones are those crucial structural proteins. Acetylation tends to loosen the chromatin structure, making the DNA more accessible for the cell's machinery to read and transcribe genes.
So acetylation is more like turning the volume up.
Often, yes. And deacetylation, the removal of those groups, condenses chromatin, making genes less accessible, typically silencing them.
Got it.
So it's absolutely critical to understand that these epigenetic changes are not alterations to the underlying DNA sequence itself. That stays constant between the twins.
Okay, that's key.
Right. These are modifications on or around the DNA that profoundly affect how those genes are expressed, turned on, off, or how loudly they're expressed.
So the musical score is the same, but the annotations, the dynamics change.
Perfect analogy. The epigenetic marks are the annotations, the tempo changes, the accents, the cues that dictate precisely how that music is played. A famous study on older twin pairs found they had remarkably different levels of these epigenetic marks.
Really? A big difference.
Strikingly so. And these differences correlated directly and powerfully with different gene expression profiles. In stark contrast, very young twins showed minimal, almost negligible divergence.
Wow. So life experience really does leave a mark, literally.
It underscores the profound and cumulative impact of lived experience on our cellular instruction sets. Environment literally annotates our genetic score.
So beyond the molecular details, what does this epigenetic drift actually mean for the twins themselves, for their health, their longevity in the real world?
Well, this divergence directly contributes to what scientists call phenotypic discordance.
Phenotypic discordance, meaning they look or function differently despite the same genes.
Exactly. It's when two genetically identical individuals exhibit strikingly different observable traits, health outcomes, or susceptibility to disease. Imagine one twin developing, say, type 2 diabetes or Alzheimer's.
While the other stays healthy.
Precisely. While their identical twin counterpart remains robustly healthy, sharp, active, even into their senior years. Despite sharing that identical genetic blueprint, they're differing epigenomes, shaped by unique life experiences, exposures, diet, stress.
It's these remarkably different health trajectories. It's incredible.
And the profound insight here is that over the adult lifespan, these unique environmental factors outweigh shared genetics in shaping these methylation differences.
Outweigh genetics, that's a huge statement.
It is. It's a critical, almost paradigm-shifting understanding. It signifies that while your starting genetic hand is fixed, the cards you're dealt at birth, how you play that hand,
Do lifestyle, choices, environment?
dramatically impacts your long-term health, your disease risk, your health span. It fundamentally challenges strict genetic determinism.
So it puts a lot more power, more agency back in our hands.
Absolutely. You're not just a passenger on a predetermined genetic ride. You are, in a very real sense, the driver, or at least the co-pilot, of your epigenome.
This insight, this phenotypic discordance, it leads directly to understanding that our epigenome is remarkably plastic.
Exactly, plastic. It's not rigid or static. It's highly adaptable, responsive, constantly evolving. It's the interface between our fixed genome and our dynamic external world.
So things like diet, smoking, stress, exercise, even toxins.
All of them. They can leave distinct, lasting marks on your DNA and histones. And as the twin studies show, these marks diverge between individuals and profoundly influence their aging trajectories.
It's a powerful idea, offering that sense of agency over our biological destiny. Our choices literally inscribing instructions.
And it's crucial to note these epigenetic alterations are now widely recognized as a fundamental hallmark of aging.
One of the core pillars of why we age.
Indeed. As we age, there's a typical complex pattern. Generally, a global DNA hypomethylation and overall decrease across the genome, like losing some off switches.
Okay.
But simultaneously, site-specific hypermethylation of certain gene promoters.
So turning off genes that should be on, like repair genes.
Potentially, yes.
Yeah.
Leading to inappropriate silencing. We also see significant shifts in histone modification patterns, changes in chromatin accessibility.
So the whole structure changes.
In subtle but important ways. For instance, in premature aging syndromes, progeria, we often see a loss of heterochromatin marks those associated with tightly packed, silenced chromatin.
Like H3K9E3.
Exactly, H3K9Me3, H3K27Me3, and altered histone acetylation. These changes strikingly mirror normal aging.
Suggesting a common underlying mechanism.
Precisely. And the overall chromatin structure tends to relax with age, which paradoxically can cause problems.
Like what?
Like activating transposable elements, jumping genes.
Uh-oh.
Yeah, they can insert themselves elsewhere, leading to genomic instability, disrupting gene function. So this multifaceted epigenetic drift, DNA methylation, histone marks, chromatin accessibility is truly central to the biology of aging.
And the twin data show that while some of this is random,
a significant portion is lifestyle mediated, and therefore potentially modifiable.
which is the hopeful part, right? If
Yeah.
we can modify these marks through choices or therapies.
We might genuinely be able to slow down detrimental aspects of aging and extend not just lifespan, but crucially, health span.
Okay, let's do a quick micro recap here. Identical twins start almost identical epigenetically,
Right.
but unique life experiences cause their epigenetic landscapes to drift apart over time.
Measurably so.
This directly influences their health outcomes, disease risk, their whole aging trajectory.
Absolutely.
Underscoring this remarkable plasticity of our epigenomes, it's not just theory, it's observable biology.
And it shifts our understanding away from pure genetic determinism.
Towards a more dynamic view where our choices really matter at a deep biological level.
Precisely. B DNA methylation clocks. Markers versus drivers.
Now, this is where it gets really interesting and maybe a little mind-bending. Epigenetic clocks. It sounds like science fiction. How do scientists actually read such a clock?
It is pretty astonishing. It's one of the most significant breakthroughs in geroscience, the study of biological aging.
So not just curiosities.
Far from it. These are sophisticated algorithms. They analyze incredibly precise patterns of DNA methylation at specific CPG sites.
Those cytosine-guanine pairs where methylation happens?
Exactly. By analyzing patterns across hundreds, even thousands of these sites, the algorithms estimate an individual's biological age.
So not just birthdays, but like biological mileage, wear and tear.
That's a great way to put it. Biological odometer. It tells you how much metabolic mileage your system has accumulated.
Are there different types of these clocks?
Well, yes. Prominent examples include Horvath's clock, one of the earliest, works across many tissues. Then there's PhenoAge.
That one uses clinical biomarkers too, right? Like blood test results.
Correct. It incorporates things like albumin, creatinine, glucose, alongside methylation data to predict biological age more accurately. And then there's Grimmage.
Grimmage sounds ominous.
Huh? Well, it's particularly powerful. It was built using methylation markers associated with lifestyle factors like smoking, inflammation, other risks. And it shows an exceptionally strong correlation with all-cause mortality risk.
Wow. Even from a single measurement.
Even at a single time point, yes. Clocks like Grimmage and PhenoAge don't just correlate strongly with chronological age, they are powerful predictors of age-related disease risk and mortality.
So a younger biological clock age really means a longer, healthier life is more likely.
The correlation is very strong. It's becoming a profound diagnostic and prognostic tool. Which brings us to that fundamental, almost philosophical question.
The big one, marker versus driver.
Exactly. Are these clock changes simply passive markers reflecting underlying aging processes? Like a smoke detector just telling you there's a fire?
Or do the changes in methylation patterns themselves causally contribute to functional aging? Is the clock itself part of the aging mechanism?
Precisely. And it's a critical question for you, the listener, to consider. If we manage to rewind the clock, make methylation patterns look younger.
Are we truly slowing aging fundamentally?
Yeah.
Or just changing the readout, like resetting the odometer without fixing the engine?
That's the crux of it. The debate has been intense, but evidence is increasingly pointing towards a causal role.
What kind of evidence?
The most compelling comes from cellular reprogramming. Remember the Yamanaka factors?
OSKM, the ones that make induced pluripotent stem cells.
Exactly. If you partially express those factors OCT4, SOX2, KLF4, c-MYC in mice, you can literally rewind their methylation patterns.
Just change the clock number.
It goes beyond that. It's been shown to improve tissue function, effectively reversing some visible and measurable signs of aging in these animals.
Whoa, so the changes aren't just cosmetic, they actually reset function.
That's what the evidence strongly suggests. It demonstrates that epigenetic changes aren't just passive bystanders. They can actively orchestrate biological outcomes, even reverse aspects of the aging phenotype. It's revolutionary,
Right.
But, and there's always a but with powerful technology.
Caution is needed.
Absolutely. As our source notes, resetting epigenetics can be a double-edged sword.
How so?
While the potential for rejuvenation is incredibly promising, maybe one day reversing age-related diseases, uncontrolled or imprecise epigenetic changes could lead to severe unintended consequences.
It's like cancer, or cells losing their identity.
Exactly. Imagine cells losing their specialized function, or worse, contributing to uncontrolled growth. It highlights the extreme precision and deep understanding required.
So it's not just hitting a big red reset button.
Not at all. It's about meticulously fine-tuning a complex, delicately balanced symphony of gene expression, making sure only beneficial changes happen while maintaining cellular integrity.
So essentially, this DNM age, the methylation age of our cells, it acts like a cumulative record, a biological journal of our exposures and damage.
A very detailed record, yes. Meticulously recording every significant event, every stressor, every repair attempt.
And periods of extreme stress, bad diet, they can accelerate it, literally age your cells faster.
They can. Demonstrably so.
Okay.
Conversely, healthy lifestyles, regular exercise, good nutrition, stress management correlate powerfully with a younger methylation age.
Reinforcing those twin study findings again, our choices impact the biology. Rewrite that journal.
Precisely. And researchers have even noted that the epigenetic age of tissues can predict lifespan better than some traditional risk factors.
Better than cholesterol or blood pressure sometimes.
In some studies, yes. Underscoring its utility as a powerful biomarker for overall health and longevity. Is rapidly moving beyond just scientific curiosity. But despite this predictive power, that moving the clock dilemma persists. It's a central question.
Because changing the clock number doesn't always mean better health outcomes in trials.
Exactly. Trials where interventions did change DNM age have sometimes yielded unclear functional benefits. There was a small 8-week pilot study, for instance. Middle-aged men.
What did it find?
An intriguing result. A measurable 3-year reduction in Horvath's clock age in the intervention group compared to controls.
So it is possible to change the clock with lifestyle. It's malleable.
Unequivocally, yes,
Yeah.
even in the short term. But crucially, no significant clinical or functional outcomes were measured in such a short study.
So we don't know if those men were actually healthier, stronger, sharper.
We don't know from that study. It leaves us with that fundamental question. If we move the clock, does it translate to meaningful health improvements? A real reversal of physiological decline? Or is it just cosmetic at first?
So how do we bridge that gap between the clock number and actual function?
Well, scientists are developing and using newer measures, like dynamically pace.
Dundon pace. What's different about that?
Unlike static clocks giving one biological age, Dundon pace gauges the pace or rate of aging, the speed of physiological decline across multiple organ systems.
So it's more dynamic, tracking how fast you're aging, not just how old you are biologically.
Exactly. The aim is to link methylation changes more directly and robustly with actual functional aging, moving beyond just a number to a measure of active physiological decline, the health span trajectory. It's a significant step.
Nice.
All right, let's take a moment to really consolidate this complex topic of epigenetic clocks. It's a rapidly evolving field. How about a mega recap?
Good idea. Hit me with it.
Mega recap. Okay, so epigenetic clocks, they're powerful algorithms using DNA methylation patterns to estimate biological age.
And they strongly predict disease risk and mortality.
Like Grimmage.
Right. Initially, they were mostly seen as passive markers, just reflecting aging. But emerging evidence, especially from reprogramming experiments with the Yamanaka factors,
Suggests they might actually causally contribute to aging phenotypes. Influencing them could impact how we age.
correct. However, the precise link between rewinding these clocks in humans and getting tangible, clinically significant health improvements, that's still under active investigation. We need more data from longer, larger studies.
Absolutely. And newer tools like Dune and Pace are aiming for a more functional assessment, focusing on the pace of aging, trying to forge that undeniable link between epigenetic changes and real-world health outcomes.
So it's a field in constant motion, constantly refining our understanding.
Definitely. Now, maybe a quick flashcard list to really nail down the key terms.
Yeah, let's do it.
Flashcard bullet list. First up, epigenetic clocks. Algorithms using DNA methylation at CPG sites to estimate biological age.
Got it.
Examples: Horvath's, phenoage, Grimmage. They predict disease risk and mortality.
Check.
The big debate, marker versus driver. Do changes cause aging or just reflect it?
Okay.
Causal evidence hint. Partial Yamanaka factors, OSKM, can rewind methylation and improve function, at least in mice.
Right, the reprogramming data.
DNAM age. Reflects cumulative exposures, accelerates with stress-poor diet, slows with healthy lifestyle.
Makes sense.
Moving the clock. We know methylation is malleable, like that 3-year reduction pilot study, but the functional benefits in humans are still unclear.
The translation gap.
And finally, Dindon pace, a newer measure tracking the pace of aging, aiming to link methylation more directly to physiological decline.
Excellent summary. Really helps clarify the key points. #2, sirtuins, NAD+, and the CD38 tug-of-war, epigenetics and metabolism crosstalk. Okay, so we've established this dynamic nature of epigenetics, this interface between genes and environment. But, the question remains, how does a cell actually know when to make these changes? How does it sense its environment, its energy status, its metabolic health?
That's the crucial link. How are those external signals translated into internal epigenetic modifications?
Exactly. And this is where epigenetics doesn't happen in a vacuum, right? It crosstalks profoundly with cellular metabolism.
Absolutely. And a central molecule in that crosstalk is NAD, nicotinamide adenine dinucleotide.
NAD. It seems to pop up everywhere in longevity discussions.
It's fundamental. Think of it as a vital coenzyme, yes, but also like the cell's internal communication network, signaling the energetic state of the whole system. It's involved in hundreds of essential reactions, from making energy in mitochondria to repairing DNA, the role of sirtuins. And this brings us directly to a remarkable family of enzymes. The sirtuins, SIRT21 through SIRTCAS7.
Ah, the sirtuins. Also big players in aging research.
Critically important NADO-dependent enzymes, meaning they absolutely require NADU to function. No NADU, no sirtuin activity, like an engine without fuel.
Okay, so NAD+ is their essential cofactor. What do they do?
Their primary role is to deacetylate histones and other important non-histone proteins.
Deacetylate, remove those acetyl groups we talked about earlier.
Precisely. When sirtuins remove acetyl groups from histones, it causes the chromatin structure to condense, typically leading to gene silencing, turning genes off.
But they can activate things too.
In other contexts, yes, they can deacetylate and activate transcription factors that promote gene expression. The key point is, this process creates a direct, elegant link.
Between what?
Between the cell's nutrient status, reflected by NAD availability, which mirrors the cell's energy and redox state directly to gene expression via these chromatin modifications and protein regulation.
So NAD+ levels tell the sirtuins how to tweak gene expression based on energy.
It's a fundamental mechanism for cells to rapidly adapt their genetic output based on nutrient availability and energy demands. Incredibly flexible.
And this is where sirtuins get really exciting for longevity, right? Calorie restriction.
Exactly. In calorie-restricted animals, still the gold standard for extending lifespan in labs.
Across many species, from yeast to monkeys.
Right. In those animals, SIRT1 activity is consistently and significantly upregulated. Its levels and activity increase.
And this isn't just a correlation, it actually contributes to the longer lifespan.
The evidence strongly suggests it does. Partly by dialing down IGF-1, signaling that growth pathway associated with aging, and by enhancing genome stability.
Keeping the DNA intact, like cellular librarians and repair crews.
A good analogy. Preserving the integrity of the genetic information. And likewise, overexpression of SIRT6, another sirtuin.
What does SIRT6 do?
It's been shown to extend mouse lifespan, significantly in males at least, and powerfully improves crucial processes like DNA repair fidelity and telomere maintenance.
Telomeres, the protective caps on chromosomes that shorten with age.
The very same. Boosting SIRT6 helps maintain their integrity, ensuring genomic stability.
So these findings strongly suggest that boosting sirtuin activity, maybe by increasing NAD, could be a powerful strategy.
It could represent a promising way to slow specific detrimental aspects of aging, turning down the dial on cellular decline, promoting resilience. The therapeutic potential is remarkable. See, the NAD plus decline with age.
Okay, this sounds incredibly promising. A clear path. Boost NAD, boost sirtuins, slow aging. Simple, right?
If only were that simple. Here's the fundamental challenge, the critical roadblock.
What is it?
NAD levels naturally decline with age in many tissues throughout the body, and it can be quite a significant drop.
So the fuel for sirtuins just disappears. Why? What's draining this vital coenzyme, this cellular currency?
It's like the cell's central battery is slowly losing its charge, and we need to understand the leak. It's a sustained depletion. D, the CD38 tug of war. And a major, increasingly recognized reason for this age-related NAD decline is the activity of an enzyme called CD38.
CD38. Okay, what does it do?
It's a potent NAD-consuming enzyme. It literally breaks down and uses up NAD.
So it's actively depleting the NAD plus pool.
Think of NAD as water in a crucial cellular pool. CD38 is like a very active pump, constantly drawing water out.
And the problem gets worse with age.
Yes, the expression and activity of CD38 rise significantly with age, especially in inflammatory conditions, which are also characteristic of aging, that whole inflammaging concept.
So as we age, the CD38 pump gets more active, draining more NAD.
That's the picture, making it harder for the cell to maintain optimal levels. This increased CD38 activity drains the NAD tool, directly and severely impairing sirtuin function, because sirtuins need that NAD.
Creating this tug of war, you meant.
Exactly. NADO and sirtuins representing pro-longevity pathways battling against age-associated depletion driven by CD38, a metabolic arms race within our cells.
Is there good evidence for CD38's role?
Very compelling evidence. Studies in mice genetically engineered to lack the CD38 enzyme.
What happens to them?
They are protected from age-related NAD loss, and they show remarkably better mitochondrial function, the cell powerhouses.
Wow. And what about inhibiting CD38?
Even more striking, giving old mice a potent CD38 inhibitor, a compound that blocks its activity, has been shown to restore NAD levels and metabolic health to a more youthful state.
That's huge. Strong evidence for CD38 as a major antagonist to healthy aging, a key target.
It certainly appears to be a key player in the age-related decline of cellular vitality. NAD plus augmentation strategies.
Okay, so given this crucial role of NAD and its decline, driven partly by CD38, augmenting NAD has become a really hot topic, right?
A major focus in longevity research and potential interventions, yeah.
Using NAD precursors, like NR or NMN.
Nicotinamide riboside, NR, or nicotinamide mononucleotide, NMN, exactly. They're compounds the body can use as building blocks to synthesize more NAD, aiming to replenish those declining levels.
And they've shown promise in animals, improving cell function, extending lifespan sometimes.
They have shown significant promise in various animal models, yes. The fundamental idea is simple. Replenish the drained NAD pool, restore sirtuin activity, support all NAD-activated processes, human trials, mixed outcomes. However, when we move from animal models to human clinical trials,
the human translation challenge again.
The early phase results for NAD boosters present a more nuanced kind of mixed outcomes picture.
Meaning they raise NAD plus levels, but the health benefits aren't always clear.
Precisely. They can safely raise NAD levels in humans, which crucial proof of concept. And sometimes they show modest improvements in specific metabolic markers, say in muscle or liver.
But the impact on tangible aging outcomes, still unclear.
In the shorter-term human studies, yes. Some trials report encouraging, modest improvements, better muscle endurance, slight inflammation reduction. Positive signals.
But others find no significant clinical change beyond just raising NADO.
Correct. No clear, widespread, robust improvement in age-related health issues observed consistently across all trials yet.
So why does NAD plus repletion remain such an appealing strategy then?
Because of its fundamental pervasive role in so many critical cellular processes. DNA repair, mitochondrial function, sirtuin activity. It touches almost everything. It's like replenishing a foundational resource for the whole cellular ecosystem.
But we still need the results from bigger, longer human studies to prove a real anti-aging effect.
Absolutely. We need studies measuring hard clinical endpoints related to healthspan and lifespan, not just biomarkers.
So in the meantime, while the jury's out on supplements,
supporting your own intrinsic NAD system through proven methods, regular exercise, a healthy diet is a prudent and highly accessible approach.
Because those things can upregulate the body's natural NAD plus recycling enzymes.
Exactly. They help your body become more efficient at making and recycling NADU naturally, optimizing the internal system, gene nutrient sensing pathways, steer epigenetics. And this whole discussion around NAD plus and sirtuins really highlights a bigger principle.
Which is?
That nutrient-sensing pathways and various metabolites fundamentally regulate epigenetic enzymes. It's not simple cause and effect, it's a symphony.
Where the cell's energy state and redox balance
directly influence chromatin modifications and epigenetic changes, which then dictate gene expression.
Can you give another example?
Sure. The metabolite acetyl-CoA is a classic one. When nutrients are abundant, acetyl-CoA levels are high. This can increase histone acetylation.
Opening up chromatin, increasing gene transcription.
Generally, yes. Conversely, scarce nutrients mean low acetyl-CoA, reduced acetylation, often suppress gene expression. This synergy allows cells to rapidly reprogram gene expression in response to nutrient availability.
Which is critical for stress resistance, resilience, and longevity.
Absolutely. This continuous feedback loop allows cells to optimize function based on the immediate environment, influencing long-term health.
Okay, quick micro recap for this section. The NAD plus sirtuin system is a vital link between our metabolic state and epigenetic regulation.
The crucial connection point.
As we age, CD38 activity increases, sapping our NAD plus sac, hindering sirtuins.
A key part of the age-related decline.
Boosting NAD plus with precursors like NRNMN is a compelling idea, shown promise in animals.
But human results are still emerging, somewhat mixed so far.
Right, reminding us that supporting our natural NAD plus systems through lifestyle exercise, diet is paramount right now.
Optimizing the intrinsic machinery is always a good foundation. #tag3. Nutrient sensing pathways steer epigenetics, continued. A deregulated nutrient sensing is a hallmark. And building on that idea of intrinsic machinery, moving beyond just NAD plus and sirtuins, aging is also profoundly characterized by deregulated nutrient sensing.
Not just a side effect, but another fundamental hallmark of aging.
Indeed. And it directly converges with and drives epigenetic change. Think of it as the cell's master control system for perceiving nutrients and regulating longevity.
Like a sophisticated chemical sensor that gets less precise over time.
Exactly, almost deaf to the nuanced signals about nutrient availability. This misinterpretation leads to suboptimal responses, bad resource allocation, failure to activate protective pathways.
Which drives age-related decline, like insulin resistance.
Precisely. Insulin resistance, altered mTOR activity, impaired AMPK activation, all contributing to a less resilient cellular state. B, the longevity nutrient sensing network.
Okay, so if deregulated nutrient sensing is a hallmark, what are the core components of this system, this longevity nutrient sensing network?
It's a complex, interconnected web, the cell's central command for energy management. 3 major players stand out.
Let's hear them. First, insulin IGF-1 signaling. I say, this responds quickly to nutrients and growth factors, especially glucose and amino acids.
Yes, it's the cell's alarm system for nutrient abundance. High insulin IGF-1 signals plentiful resources, time to grow and divide. But what's consistently fascinating, maybe counterintuitive,
is that reduced ICE activity is linked to longer lifespan.
Robustly and consistently across many organisms. Think of C. elegans, that little research worm. Specific DAF2 mutants with dampened IC live significantly longer, sometimes more than twice as long.
Wow. And similar findings in mice.
Yes, certain long-lived mouse models involve reduced ICE activity, genetically or pharmacologically.
How does lower ICEs help?
It's linked to activating stress response genes, helping cells cope with damage and better maintenance of adult stem cells, crucial for tissue repair.
And the epigenetic connection.
ICES powerfully modulates FOXO transcription factors.
Master regulators controlling genes for stress resistance, DNA repair, metabolism.
Exactly. By influencing FOXO activity and location, ICES doesn't just affect metabolism superficially. It directly influences the transcriptional profile of cells, partly via downstream effectors that alter chromatin structure.
The profound connection between a metabolic signal and the gene expression machinery.
Absolutely. And intriguingly, human centenarians, people living to 100 plus, often carry specific genetic polymorphisms that naturally dampen ICES.
Suggesting lower ICEs might predispose humans to extreme longevity too. A glimpse into exceptional aging.
It certainly suggests that.
Okay, second player, mTOR. Mechanistic target of rapamycin, another crucial nutrient sensor.
Yes, senses nutrient abundance, especially amino acids and glucose, and energy status. When things are plentiful, mTOR activates.
Promoting growth, protein synthesis, cell proliferation, the go-go-go signal.
That's a good way to think of it. Conversely, inhibiting mTOR, maybe with the drug rapamycin or just through nutrient scarcity,
is a powerful way to extend lifespan.
Widely demonstrated in many organisms, yes. mTOR influences epigenetics by orchestrating vast gene expression programs related to metabolism, protein synthesis, even autophagy, the cell's cleaning process.
So, in the third player, AMPK. AMP-activated protein kinase, the cellular energy sensor, the fuel gauge.
One of the most fundamental, yes, it monitors the cell's energy levels. When energy is low, high AMP-ATP ratio, like during exercise or calorie restriction, AMPK activates.
And when active, it flips a switch, promotes energy production, inhibits energy consumption.
Exactly.
Yeah.
Promotes catabolic processes, making energy glucose uptake, fat burning, and inhibits anabolic processes, using energy protein synthesis, fat storage, the cell's crisis manager, C, interplay and synergy. And the connection to epigenetics here is direct and profound too. AMPK can directly phosphorylate histone-modifying enzymes and transcription factors.
So it directly influences chromatin and gene expression, telling the cell to conserve energy and shift metabolic priorities.
Correct. And the key takeaway really is the interconnectedness. IS, MTR, AMPK, they aren't isolated. They form a complex, integrated, dynamically regulated network.
Constantly talking to each other.
Their signaling directly impacts epigenetic enzymes, like sirtuins, influences chromatin structure, allowing cells to dynamically adapt gene expression in response to nutrient availability, metabolic stress, environmental changes.
Ultimately impacting the whole aging process. It's like cellular intelligence adapting the genetic landscape to the environment.
Ensuring resilience and adaptability, yes. A, caloric restriction in humans. Calorie findings.
Okay. Given this profound impact of nutrient sensing on everything from gene expression to longevity, it brings us naturally to caloric restriction, CR.
Consuming fewer calories while maintaining adequate nutrition.
For decades, this has been the gold standard intervention to slow aging in lab animals, right? Consistent lifespan extension across species.
From yeast to monkeys, yes. It's been a cornerstone of aging research, fueling hope for human translation. And that human translation has been a major focus. The calorie study, comprehensive assessment of long-term effects of reducing intake of energy.
The most rigorous human trial of CR, over 2 years.
That's the one. And it meticulously showed that many of those benefits do indeed translate to people. A significant finding.
What kind of benefits?
Clear improvements in blood markers from metabolism and inflammation, lower fasting insulin, better insulin sensitivity, lower C-reactive protein, and even more intriguingly,
a slowing of aging biology.
A slight yet statistically significant deceleration of aging biology as measured by certain biomarkers. Things like slower metabolic rate, decreased oxidative stress.
So not a magic bullet, but proof that the fundamental biology is conserved in humans. We can influence our aging rate.
It powerfully demonstrated that, yes. The underlying mechanisms involve activating those pathways we discussed, AMPK, sirtuins, and inhibiting MTR and ISI, shifting the cell from growth to repair and maintenance, BCR mimetics.
However, and this is the big real-world challenge, true caloric restriction is incredibly hard to maintain.
It demands extraordinary discipline, meticulous planning, constant awareness. Most people find it exceptionally difficult long-term.
Plus potential risks. Muscle loss, bone loss, nutrient deficiencies, if not managed carefully.
Those are real concerns, yes. Which naturally leads to the exciting search for alternatives. Indeed, the formidable challenges of sustained CR have spurred intense interest in CR mimetics.
Substances or interventions mimicking the benefits of CR without the drastic calorie cutting.
That's the elegant idea. Activate the same pro-longevity pathways or inhibit the pro-aging ones, but through a more manageable approach. The source lists some examples.
Like metformin, the diabetes drug.
Metformin is a prime example. Widely prescribed, getting lots of attention for anti-aging potential. Thought to work partly by activating AMPK, mimicking some CR effects. It's being studied in large trials like TAME targeting aging with metformin.
What else?
Acarbose is another. It slows carbohydrate digestion, reducing glucose spikes, mimicking some metabolic effects of CR, potentially dampening AYA. Animal studies look promising.
And 17-estradiol, a non-feminizing estrogen.
Right. Being investigated for metabolic benefits, especially in aging males. Mechanisms aren't fully clear, but it seems to influence nutrient-sensing pathways in a CR-like way, improving health span in animal models.
So these mimetics often work by hitting those same nutrient-sensing pathways, AMPK, MTOR, ISA, or affecting metabolic processes that indirectly impact epigenetics and longevity.
They're like molecular keys designed to unlock the same beneficial cellular responses triggered by CR.
But through a potentially more manageable pharmacological approach. The hope is to get the health span benefits without the huge lifestyle burden.
Exactly. Potentially offering a more accessible path to healthier aging for more people.
#outro.
Wow. We've journeyed through this incredibly intricate, constantly evolving landscape, epigenetics and aging, from those compelling twin studies showing the power of environment,
to the sophisticated molecular clocks tracking biological age.
and that critical tug-of-war between our metabolic state, NAD plus ADCD38, sirtuins, and our longevity genes.
It's a lot to digest. It really is. But what emerges, I think, is this powerful, almost liberating understanding. While our genetic blueprint is set,
Our epigenome is not static.
Right. It's dynamic, responsive, a continuously updated interface between genes and environment, constantly being sculpted by our daily lives, diet, stress, choices.
It's not abstract theory. It's active biology happening in our cells, influencing our health trajectory. So to crystallize this for you, our listener, let's distill the key takeaways, these profound insights.
Let's hear them.
First, epigenetic plasticity is paramount. Your lifestyle profoundly influences your epigenome, making you biologically distinct even from an identical twin over time.
Remodeling gene expression, impacting health span. Our choices are powerful determinants.
Second, biological age versus chronological age is measurable and meaningful. Epigenetic clocks offer that window into biological age, which choices can accelerate or decelerate.
And while the marker versus driver debate continues, the potential for interventions is immense, maybe even reshaping our biological timeline.
Third, metabolic crosstalk is a core regulatory hub. The NAD plus sirtuin system is that crucial link between nutrient sensing and epigenetic regulation. Its age-related decline, partly via CD38, is a key vulnerability.
Understanding that tug-of-war is vital for finding intervention targets.
Fourth, nutrient sensing pathways are master orchestrators. IS, mTOR, AMPK act as master switches, guiding epigenetic changes based on energy status. They dictate cellular priorities, growth versus maintenance, critical for aging.
These pathways are fundamental control systems.
And finally, intervention potential is expanding and promising. Caloric restriction and its mimetics offer increasingly viable strategies to positively influence these pathways in epigenetics,
Hinting at a future where we can actively modulate aging, extending health span. Research is moving fast here.
from observation to potential therapeutics.
So what does this all mean for you, the discerning learner, navigating this complex world, seeking to understand and maybe influence your own trajectory? If our epigenome is so profoundly plastic, this living reflection of our cumulative exposures and choices, what new responsibilities, what incredible opportunities does this understanding present for personal health span? Does it fundamentally shift that old nature versus nurture debate?
Suggesting maybe a far more integrated dynamic interplay.
Exactly, where personal agency, conscious decisions, daily habits play a much greater, more direct role than we previously imagined, shaping not just our health, but the very rate at which we biologically age.
It's a profound thought to carry forward.
We invite you to continue exploring the implications of this deep dive into the architecture of longevity and the remarkable adaptability of your own biology.